This is a 1308 web series videos. This is chapter 3, the molecules of life. Now, as we noticed in chapter 2, chemistry is a big deal in biology. So this chapter here continues the thought of chapter 2 on how biology and chemistry are related. So we're going to start off with the first slide. Okay. So here on this slide we see a bunch of elephants, and notice it says it all has to do with the DNA. DNA and other cells of our body use certain chemistries, the molecules of life, like DNA. So we're going to see again the four main elements that I mentioned in chapter 2, which would be the carbon, the hydrogen, the oxygen, and the nitrogen. So we're going to see how those play a role in this chapter. Now, the first example that they're giving here is milk sugar, which is lactose. So lactose is your milk sugar. Again, we're going to see that these are fundamental molecules of life. And here we're starting with a carbohydrate, and the carbohydrate in this example is lactose, which is milk sugar. Now, there are some people on the planet, usually Asians, have this problem where they have a missing enzyme. The missing enzyme is called lactase. So lactase is the enzyme that breaks down lactose. And if you don't have this enzyme lactase, then it causes all kinds of fermentation going on, bloating, gas, diarrhea, all that, because you have a problem called lactose intolerance. And that's because you're missing this enzyme called lactase. So as you can see, very similar to what was mentioned before, that there's a certain structure, and if you don't have that structure, then it changes the function. So in people with lactose intolerance, they have the wrong format, the wrong structure of their enzyme called lactase, so therefore they're not able to break down milk sugar lactose. Next slide. And here's just a picture of a kid eating ice cream. And now we have here on this slide an important one that says organic compounds. By definition, organic compounds are the ones that have carbon base. So carbon-based molecules are called organic. And you can see the line here at the bottom says organic compounds are carbon-based. And they're very, very complex. Now there's something I mentioned in chapter 2 that I'm now repeating in chapter 3. That is, changing the structure of the carbon base will change its function. A very good example that I'm going to show now, or mention now and show again in a little while, is gasoline. When you go to the gas station, they have different kinds of pumps. They have the 88 octane, 92 octane, 93 octane. Those are different levels of gas. And of course, we know it commonly as regular unleaded, premium unleaded, or super premium unleaded. Well, those are actually different levels of carbon-based gasolines, and they do play a role. Some cars are requiring the premium kind. Some can go by with the cheaper regular unleaded because the structures are different, and then they give a different amount of energy. So we're going to see that in a moment. Here's a next slide showing you how carbon is very versatile. That's why we have things like carbon dioxide, carbon monoxide. That's the common poison that happens when people use generators during freezing temperatures, and they use generators within the house, or they leave the car running in the house, and that causes carbon monoxide. Carbon monoxide is quite different from carbon dioxide because of the structure. And if you look at this slide here, it says carbon atoms of organic compounds can be bound with other things. And if you look at the list, you see that this list is the same list that I said about the most common elements in living things. So we have our carbon, our hydrogen, our oxygen, and our nitrogen. The CHO-N that I mentioned in chapter 2. So it's a very important thing. The next slide shows you how the carbons can be rearranged in different positions. So we have a simple structure, a double bond structure, then we have a sort of an unbranched or branched structure, and then we have a ringed structure. Now the ring structures are the ones that are important when it comes to aromatics. So many of these perfumes that are out there, they're carbon based. So anything that has a strong aroma, like gasoline has a strong aroma, turpentine has a strong aroma, nail polish. You've heard of nail polish removers that are acetone based. Well, acetone is also a ringed structure. So things with strong aromas are ringed structures, and they have carbon rings like this. Even your testosterone, estrogen, these hormones, these steroids, they're going to be ringed structures too, because they too have an aromatic property. Aromatic property. Okay, so ring structures, look for them as an aroma-based items, like steroids, perfumes, things like that. Okay, next slide. Now comes the simplest form is methane. You do want to keep that formula in mind. CH4, that's methane. Okay, that's very, very common. CH4, methane. This is the methane that reacts with the air and then becomes carbon dioxide, and that is what causes the greenhouse effect, and that's causing global warming and all of that. So as you can see here, this is CH4 methane. And as you can see down here, it even says larger organic compounds are used in such things as gasoline, even biofuels. There are some trucks now that use biofuels where they take the byproducts of waste of corn and things like that, and they actually make fuel from them. There are actually some companies using McDonald's oil, the oil that McDonald's uses to fry their food. And when the oil has already gone rancid and it's been used too much, they can actually use that fuel, that liquid, that oil to actually run engines. Okay, so that's the biofuel. Okay, here's another form of methane. As you can see that there's a structural formula, a ball and stick, and a space filling. And if you remember in chapter 2, I mentioned that the changing in this structure here, changing the structure affects its function. Okay, the author continues by showing you this picture. Here we see hydrocarbons. And again, hydrocarbons by its definition means that it has a hydrogen in it and it has a carbon in it. So if I go back a slide, you can see that this is the hydrocarbon with carbon and hydrogen. And you can see that there's octane. That's the gasoline numbers that you see at the gas station, like 92 octane, 88 octane, that kind of thing. And then we have our dietary fats, our oils, like butter, margarine, cooking oil. All of that would be the dietary fats. Again, you can see the structural difference in how they look. Okay, next slide. This is the continuation of that, carbon chemistry, showing you that there are things that you could add on to it called functional groups. And by adding functional groups on there, it changes the way it works. So we're going to see functional groups in a moment. But functional groups added on to a carbon chain is what makes them uniquely different. Okay, next slide. Now, this is a big deal because now we're dealing with living things. So in this case, we have the most common macromolecules. This is what fitness people call macromolecules: the carbohydrates, the proteins, the amino acids. Lipids is missing from this list. It should be added on. So we have carbohydrates, proteins, nucleic acids. Those are your DNA, RNA, and of course number 4 that's missing would be the lipids. So when you go read your nutrition facts on your food products, you'll see that it says nutrition facts, and then it talks about carbohydrates, proteins, and lipids. Okay, next slide. Okay, so over here we have another important comment, and that is polymers. Now, if I take a single Lego block, that would be called a monomer. So a single Lego block is a monomer. If I add another single Lego block, that would be another monomer, but when I join them together, it becomes a polymer. So 1 unit of something is a monomer. And if I add 2 or more units to it, that becomes a polymer. So your polyester pants are actually polyesters. A type of fabric is, of course, artificial. Polyester pants are artificial. And there are many, many esters put together to give you your pant fabric, which is polymers. Now, how do I join them together? I have a process called dehydration. And that's where I'm putting the two monomers together. And notice in dehydration, I actually remove a water molecule. We'll see a picture of that shortly. Okay. So when I want to join things together, and that's, by the way, true for any of the nutrients. Let me backtrack on a slide. So this list right here that I gave you a moment ago, carbohydrates, proteins, the DNA, RNA, nucleic acids, and number 4, the lipids. How do I make these? I join any of these together by way of dehydration, which is when I take two monomers put together, and when I take two monomers or 2 or more monomers put together, that's called a polymer, and that's done by dehydration, which will remove a water molecule. Now the next paragraph says the opposite. How do I break things apart? Well, if I have a lot of long chains of stuff, which are my polymers, like long chains, how do I break them apart? I add water, and that's what the line says right here. I add water, and that process is called hydrolysis. So hydrolysis is adding water, and it breaks the polymer apart. So we're going to see a picture of these two in a moment. So polymers are made by dehydration, and polymers are broken apart by hydrolysis. Okay, so that slide's important. Here's the picture version of just the same thing. So here we have a monomer and another short polymer, and we want to add them together. We want to put them together. And in doing so, the process is called dehydration, and it forms a polymer, and notice it also creates or removes a water. So it creates and removes a water. That's dehydration. Then to break it apart, here we have a picture of a long chain, and we want to add water, and that's hydrolysis, and that breaks it apart. And that's true for all of those nutrients I showed before. Okay, next slide. So here we have again, and notice this time the author did the right thing by adding lipids to the list. Remember I said that the author had made a mistake by not adding lipids. So now you can see that lipids have been added to this list. Okay. Now we're going to talk about just each one of these individually. So each of these macromolecules, these nutrients, one at a time. So the first one is carbohydrates. And carbohydrates is really putting the simple sugars together. So the simple sugars together form carbohydrates. And it turns out that the number one purpose of carbohydrates is energy. Now, if you recall, in my earlier videos, and this was in chapter 1 and probably even in chapter 2, I mentioned that the overall body uses glucose for energy, but the cell uses ATP for energy. And glucose happens to be the simple carbohydrate. So the number one function for a carbohydrate is energy. Okay, so the number one function for a carbohydrate is energy. The number 2 function for a carbohydrate is structure. And I'm going to go ahead and give it to you for the purpose of the video. There's an example for the number 2 function, which is structure, and that's something called chitin. C-H-I-T-I-N. Chitin. It's the shell of some of these insects that you see out there. So if you've ever seen a cycad dry up and molt on a tree, well, that shell that they leave behind is made out of chitin. C-H-I-T-I-N. And it turns out to be the same type of material that's now used for those dissolvable sutures that surgeons use, where you don't have to get the sutures removed, they dissolve into your body. It's made out of the same material, chitin, CHITIN. So carbohydrates, number one function is energy, number two is chitin or structure. And again, when I say that the number one source of function is energy, it's usually in the form of glucose. Okay, next slide. So here we have monosaccharides. Monosaccharides are simple sugars. Okay, they are simple sugars. So glucose is an example of monosaccharide. Fructose is an example of monosaccharide. So in this picture here, this slide, you see glucose found in our sports drinks, and we see fructose found in fruit. So even if you're a diabetic and you're not supposed to have glucose, there are some people who overdo it by having too much fructose. And fructose does get converted to glucose in the body. So if you're a diabetic, you should not be eating too much fruit. Now here in the slide, we see that fructose and glucose are isomers. Now an isomer is just a variation of the same chemical formula. So glucose and fructose are very similar. And that's why when people argue, well, I'm not taking a lot of glucose, well, that's not true. If you don't take a lot of glucose, but you're taking a lot of fructose, it's almost the same thing. Yeah, structures are slightly different, but the formula is the same. And in the body, fructose will be handled like glucose. Okay, next slide. So in this picture, you clearly see the difference. The formulas are the same. So you see down there? So the formulas are the same. Only the chemical rearrangement is different. So even though they have the same formula, they have a different arrangement, they are different structures overall, but one way or the other, our body will eventually convert fructose to glucose, the number one energy source for a carbohydrate. Okay, moving on. Okay, I don't care for this slide. And notice the author says exactly what I told you. Monosaccharides, especially glucose, the main source of fuel for the body. And then the body converts glucose to ATP within the cell. So the cell uses ATP. Okay, next slide. Here's what a simple glucose looks like. Now on an exam, you will not be asked to draw any of these. But this last one down here, this green one, you should identify that. So when you see a hexagon shape, you know that this is a glucose, a carbohydrate. That they will ask you. Okay, next slide. Now, remember I said earlier, each one of these nutrients can be put together? Well, that's exactly what this is. We now have lactose, maltose, sucrose, which is our table sugar. Right? That's our table sugar. All of these are disaccharides. And if you remember what I said earlier, how do I put the individual monomers together? By dehydration. So by dehydration. So all of these are put together by dehydration, just like we saw in the previous slide. So disaccharides are actually two monosaccharides or two monomers put together. The high fructose corn syrup discussion down here, That's also the same thing. And of course, it's going to be handled just like fructose in the body, eventually converting it back to glucose. So that's why high fructose corn syrup is like super concentrated glucose, and that's why you gain a lot of weight and raises your blood sugar very rapidly. So the bread that I take, for example, outright says no high fructose corn syrup, which is a healthier product. Okay, next slide. This is a picture version of what we just said earlier, where we have a monosaccharide, glucose, another monosaccharide called galactose. We put them together and we get lactose. Now on the test, you won't need to know the top part, but you do need to understand that lactose, maltose, sucrose, which you saw in the previous slide, they're all disaccharides. That people will ask. So let me put that slide back up again. Okay, so there it is. Lactose, maltose, sucrose. Those are disaccharides. And they're put together very similar to this, where monosaccharides through dehydration, and you can see that water is removed and they are now joined together. Okay, next slide. As you can see, the author is doing the same thing I mentioned by looking at the nutrition facts. And when you look at the nutrition labels on your food products, you can see where it says the oils or the fats, then you have the carbohydrates, and then you have the proteins. Okay, next slide. Now we have polysaccharides. Now polysaccharides are a long chain. That's why it says monomers of monosaccharides. Long chains. The first example is starch. Starch is a polymer. So starch is a polymer. So plants use starch for their structure as well as for energy. So remember how I said that for us as humans, we use glucose for energy, and I told you that monosaccharides or carbohydrates in general, the main function of a carbohydrate is energy. So that's true for a plant as well. And I said that the second function of a carbohydrate is structure. And as you can see, plants use it as a structure for their being. You know, the plants are like cellulose and starch. So that's the kind of structure. Okay, next slide. On this slide, you see a lot of the starches. So you see a starch up here, which is a polymer. You see glycogen, which is a form of polymer as well. Now, the thing you're going to need to know for the test is this. Glycogen is found in your muscles. So over here in the picture, they show the gardener's arm, and they show the muscles in the arm, and then it says glycogen in muscle tissue. So glycogen is stored in the muscles. Now, if I were to ask a question, if this were a live class and I would ask a question, the question would be, who has more muscle glycogen? I do, or say an athlete like Lance Armstrong, the bicycle Tour de France rider. Of course, someone that's an athlete and takes a lot of glycogen by taking pasta. That's why marathon runners usually have something like a pasta salad or something like that, spaghetti, the nights before the race. That way they can load up on a lot of carbohydrates, a lot of glycogen, and that way they have more energy during their race. So glycogen is an energy storage form in muscles. So glycogen is an energy storage form in muscles. And then we have our cellulose down here, which is also a polymer. Now, the difference is this. Cellulose cannot be broken down by enzymes that we humans have. So we don't have enzymes to break down cellulose. So if you eat celery and things like that, which have a lot of cellulose in them, then you're not going to break it down, and that will be the fiber that you have in your diet to help you move things through your gut. So cellulose is something that is unique, and only cows, for example, have special enzymes that they can break down cellulose with. We can't. So this is a plant wall. And as you can see here, it says cellulose plant wall. And that's why plants use cellulose type of thing as a structure. So yes, carbohydrates can be number one for energy and number two for structure. And chitin was the other example I gave earlier. So chitin is also a polysaccharide. So chitin is also a polysaccharide. Okay, moving on. So here's the author's interpretation of the same thing. I already told you that glycogen is found in the muscles. And then down here, the author says cellulose. And notice we cannot break it down because we animals do not have the enzyme to do so. And that's what adds bulk when we eat fiber plant products. Okay, next slide. Okay, this is a repeat. Remember I said that the author always shows this slide for every chapter. Same thing. Okay, now we have lipids. Now lipids are interesting. Lipids are primarily for structure. So lipids are primarily for structure. All right, so lipids are primarily for structure. So for us as humans, we really use lipids for structure. Now lipids are also hydrophilic, and the other ends are hydrophobic. And we'll see that in a minute, where I say that lipids have two ends. Hydrophilic end and hydrophobic end. Now because it has two ends, one that's hydrophilic and it's attracting water, so hydrophilic attracts water, and we have another end that's hydrophobic that is also fearing water or repelling water. So when you look at a lipid, it has two ends. One end is attracting water, the other end is repelling water. Okay? So lipids are unique. Now some people use a word that you might see called amphipathic. Amphipathic. And amphipathic simply is a word saying something that has two different ends with two different purposes. So your pencil, for example, would be amphipathic because one end of the pencil you write with, and the other end of the pencil is the eraser. So a pencil would be considered amphipathic because one end you write with, the other end you erase with. So pencils have two ends with two different purposes. Okay. So now you know that lipids' number one function, so the lipids' number one function is structure, and the number two function for a lipid will be energy. So it's the opposite of carbohydrates. So lipids' number one function is structure, and lipids' number two function is energy. Alrighty, here we go. So in this picture here, we see oil and vinegar. It could have been oil and water as well. It wouldn't have mattered. And what we have here are layerings formed because since oil has one end that attracts something and the other end that repels it, what happens is you form a layer. So here's vinegar or water, and then you have oil sitting on the top because oil has two different ends. Let me backtrack again. So if I were to take this container, this salad dressing container of oil and vinegar, and I shake it, shake, and I let it sit for a while, it's going to go back to the same layering again because it's going to orient itself in such a way that again is going to find the hydrophilic and hydrophobic and is going to form the same layering again. So you can shake this bottle as many times as you want. You let it sit and settle, and once it settles, it again forms a layering. Okay, now we do the lipids. Again, they're called fats. Now the same one here, the first one here is triglyceride. Now a triglyceride is important for two reasons. One, it's made out of glycerol, and we'll see a picture of this. It's made out of a glycerol and 3 fatty acids. Again, notice the author says by way of dehydration. So we're actually adding each of the fatty acids to this one glycerol by dehydration. Okay? So like all the nutrients, we add the pieces with dehydration, we remove the pieces with hydrolysis. Now, the author says the same thing that I mentioned, but remember, for test purposes, we're going to go with what? Fat's number one purpose, or lipids' number one purpose, is structure, and the second function is energy. Okay, so we're going to go with that direction. Number one being structure, number two being energy, the opposite of carbohydrates. And the triglyceride. Now, this is your fat energy. So in your blood, you actually have triglycerides moving around, and that is your fat energy. So your carbohydrate energy is glucose. So your carbohydrate energy is glucose, but the fat energy will be triglycerides. Okay, next slide. Okay, here's a picture of a glycerol and one fatty acid. So this would be called a monoglyceride because it has only one fatty acid attached. Again, it's done by dehydration. But if I have all three of these fatty acids attached, then it's called triglyceride. Triglyceride. Now, if you look carefully in this picture, you see that this first one up here is straight. The second one is straight. But the third one has a little kink in it. And right where the kink is a double bond. If you look carefully, you'll see a double bond right where that bend is. Now that kink or that bend, that double bond, provides it for having a little bit more fluidity, more liquid. Whereas if it's a single bond, see how these are all having one stick, one stick, one stick in between? That's more rigid. So if it's a single bond, it's rigid. If it has a double bond in it, like in this example here with the kink where the bend is, that's going to be more fluid, more liquid, more bendable, more movable. Now, why is that important? Because that's how we decide how thick, in terms of fluid, a fat is. Now, a fat can come in two forms. Now, let's look at this. If you know what Crisco is, Crisco is that vegetable shortening that you use in cooking. Well, Crisco is a vegetable shortening made out of fat. But what form is Crisco in at room temperature? So when you go in the pantry and get the Crisco vegetable shortening, it's a solid. Why is it a solid? Because all of those have straight sticks. They are solids at room temperature. So Crisco, your vegetable shortening, is a solid at room temperature. Whereas, say, canola oil or palm seed oil or any of these other cooking spray oils that are liquid at room temperature, they're going to have this double bond kink. So this double bond kink makes it a liquid at room temperature, whereas the single bonds make it solid at room temperature. So Crisco, the vegetable shortening, is going to have only the straight sticks because they're solid at room temperature, and the the Crisco, the canola oil, that canola oil is going to be a liquid at room temperature. Okay, next slide. All right. Now, this is exactly what I just said a moment ago, but now there's terms that are added to it. So when I told you a moment ago that it has at least one double bond in it, well, that double bond makes it unsaturated. So unsaturated has a double bond, which makes it a liquid at room temperature. Now you see how I'm connecting the slides here. So unsaturated is double bond, and it's a liquid at room temperature like canola oil. Then we have saturated. Saturated is the long stick I gave you with only single bonds. So saturated is the one with the single stick, and it's all single bonds, and therefore it's a solid at room temperature, like the Crisco vegetable shortening example. And so here it says if it's a saturated fat, it would have all three fatty acids being saturated, and therefore it would be like Crisco vegetable shortening a solid at room temperature. Okay, next slide. All right, here's a good example of the different oils. So you can see butter. Butter is a solid at room temperature, and therefore it is more saturated because butter is a solid at room temperature. Whereas palm oil, corn oil, or canola oil in my example, they are more what? They're more unsaturated, so they're going to be more of a liquid nature. Okay, next slide. All right. Now, I've already mentioned this. I said oils, and if you have a lot of saturated oils, and I did tell you a moment ago that saturated oils are solid at room temperature. Okay, so they're solid at room temperatures. When it comes to fish oil, that's why fish oil is considered healthy. Why is fish oil healthy? Because it's an unsaturated fat. And fish oils are unsaturated, meaning they are liquid at room temperature. So fish oils are unsaturated, they're liquid at room temperature. So as you can see, the last few slides are re-stressing the same thing. Okay, now this is what I mentioned about the Crisco, the vegetable shortening that happens to be solid at room temperature. How do they get it that way? Well, what they do is they add hydrogenated oil, which basically is adding hydrogen to it. And what it does at the factory, when they make Crisco and put them in those little tins or those little containers, they actually inject hydrogen, and in doing so, they convert the unsaturated oil to saturated, which makes it go from a liquid to a solid at room temperature. And that's why these kinds of fats are not healthy. Okay. All right, next slide. So you can see here the saturated fats are solids at room temperature. The healthy fats are unsaturated. Okay. All right, next. Okay, now we come to a variation of lipids. Lipids have variations, and one of them is what's called a steroid. Now, for the purpose of the test, remember how I said earlier steroids would have rings because they're aromatic? Remember I said that at the beginning of this one? They're aromatic. So they're going to have a ring structure. So steroids will have a ring structure. How many rings for purpose of the test? 4 interlocking rings. So steroids have 4 interlocking rings. Okay, so steroids have 4 interlocking rings. Now, one of the key examples of steroid is cholesterol. Cholesterol. Now, cholesterol is important because it makes up a big chunk of the cell membrane. So every cell of your body has cholesterol built in. So every cell of your body in their cell membrane has cholesterol built in. It plays a role in the structure of how cell membranes move. And as they say here, cholesterol is also the basic building block of steroids. So not only do cholesterol make a big part of the cell membrane, but cholesterol is also the building block of steroids. So your estrogen, your testosterone, all your major hormones, like sex hormones, tend to have steroid, and therefore they're made out of cholesterol. Now the next slide really says it best, and that's this picture here. So the top shows you what cholesterol looks like. Notice it has the ring structure because it's a steroid, and it has 4 interlocking rings. And you can see testosterone and estrogen having the steroid building blocks, the 4 interlocking rings. Okay, next slide. I don't care about this one. Don't care about that. Now we have the next type of macromolecule or nutrient, which are proteins. Proteins play the most important functional role. So if I were an in-person instructor, on the board I would be actually writing proteins are multifunctional. Okay? Because there's just too many functions to remember for a protein, so I just call them multifunctional. So anything that happens in the body, and look how the author says it, are instrumental in almost everything cells do. So if a body is doing something, then that something is going to be done because of a protein. Now, the other way I teach it is a verb. Isn't a verb an action? Like in English class, a verb would be an action word. Well, if I say words like oxygen is carried, right? That's like a verb, right? Oxygen carries. Well, if oxygen carries, well, who is carrying the oxygen? The red blood cell. So if oxygen is carried by red blood cells, that means red blood cells must be made out of what? Proteins. That's how it works. Your muscles move, right? Since move is a verb, just like carries is a verb, if muscles are moving, then muscles are made out of what? Protein. So that's how you need to remember it. Anytime it's a verb, if it does something, it has something to do with proteins. Okay, next slide. This is a list of all the things that proteins do. And this list is sometimes easily forgettable. So that's why I said keep that verb story in mind. So as you can see here, it says muscles, showing a guy with a little dumbbell there with his muscles. See how it says muscles to contract. Well, if contracting or contraction or contract is a verb, then you know it has something to do with proteins. Structure, support. Your hair, for example, has protein in it, like collagen that you may have heard of. Well, collagen is in your skin. It supports your skin. People that take all those beauty products say it has collagen in it. Well, your hair has collagen too because it's for support. Okay? Then we have things like enzymes. Don't enzymes do stuff? Don't they speed up reactions? Well, if enzymes speed up reactions, then guess what? It is made out of a protein. Earlier I said about oxygen and blood. Well, these are red blood cells here. Since I see the verb here, transports is a verb. Well, that means oxygen is transported by red blood cells, and red blood cells are made out of protein. Okay? So much easier to remember that multifunctional verb story. Okay? Now we're on what proteins are made out of. The monomer of a protein is called an amino acid. So just like carbohydrates had monomers, lipids had monomers, well now proteins have monomers, amino acids. There are 20 known amino acids. You will need to remember that number. So 20 known amino acids. Very important. 20 known amino acids. And the amino acids, how do I join amino acids? How do you think it works? Now, remember, you have to remember things along the way. So if I said all nutrients are put together, all monomers are put together in a certain way. So again, here too, if I take one amino acid and another amino acid and I want to put them together, I put them together by dehydration, just like we saw before. So dehydration, where I put the two monomers together, in this case two amino acids together, and I also form water. So we're going to see that. Now these are the three functional groups. Remember how I said that we add things on and it becomes a functional group that takes on a new ability. Well, we're going to see these in a moment, so there's no reason to write these down at this moment. Okay, so I don't care. Now this picture I do care. So here you do see those functional groups that were on the slide that I intentionally skipped. That's why I said I didn't care for this bottom part. Remember, I already told you about the amino acids. I already told you about the 20 known amino acids and how they're put together. And if you look at this picture we're on now, we see one amino acid. One end is called the amino group. That's called the NH 2. So we have an N, a nitrogen, and 2 H's. So that's NH2. And then over here on the other side, we have the carboxyl group, which is COOH. COOH. And then we have something in the yellow that says side chain. Now I'll talk about that in just a moment. Okay. So notice just this picture up the top, we see our 4 letters again. Carbon, hydrogen, oxygen, nitrogen, that CHO-N thing. Okay. So we'll see that again. Then down here, this side chain. This yellow side chain is what I call the R group. Remember we said that we have 20 known amino acids. Well, how do I come up with 20 different versions? I come up with 20 different versions by having different side chains. So by having different side chains, what I call the R group, so this yellow area is what I call the R group. Well, by having a different R group, I will come up with my 20 different amino acids. And the sequence of these chains, so if I have different colored beads, let's say I have 4 different colored beads, I can put them in a long chain in many different combinations because I have 4 different colors, correct? Well, the same thing here. When I put these amino acids together to form a long chain, the sequence of that chain is what matters. That sequence will give us a different kind of protein. There are billions of possibilities of different proteins out there. And the only way we get the different billions and billions of proteins is by having different sequences out of the 20 amino acids. Okay, next slide. So now this says the same thing I did. I just want to make sure you remembered it. How do I put amino acids together? Just as usual, by dehydration. But this time we form something special called a peptide bond. And we'll see that in the next picture. A peptide bond. And when you form a polymer, the special name for a polymer of amino acids, it's called polypeptides. So when I have a big protein, that big protein actually came from different polypeptides. Okay? So it came from different polypeptides. Okay? So different amino acids put together, and when you form a peptide bond, and you have long chains of those, we get a polypeptide. And notice the last line here, very important. Let me quickly highlight it, and then let me go ahead and talk about it. A functional protein is one or more polypeptide chains precisely, and that word is important, precisely. It's specifically twisted, folded, and coiled in a unique shape. Now, if you remember for one of my other videos of chapters 1 or 2, most likely chapter 1, I gave you something about frying an egg. When you fry an egg and you crack the egg open, the first part that comes out is that clear, runny part. That is albumin, a protein. And then when you fry it, because you're heating it up on a skillet, I told you that one of the ways that you can change a structure is by heating it up. And so the clear part of the egg, the albumin, turns white because you've done what? You've changed the shape of the albumin. And by changing the shape of the structure, it changes its function. And that's what should be added right here. By changing that unique shape to something else, it changes its function as well. So sickle cell anemia is the one I gave out earlier in a previous chapter. Well, in sickle cell anemia, instead of having a normal donut-shaped red blood cell that carries the normal amount of oxygen, in sickle cell anemia, we have now destroyed the protein. It no longer has the correct shape. That's why it's a sickle shape. And now it carries less oxygen. And therefore, the function has changed as well. Okay, the next slide is important, showing you how amino acids are joined. So we're joining them by dehydration, producing water. Notice we have different R groups. They could be the same or different. And then, of course, the sequence of those matters. And when we join the two together, we form a peptide bond. Okay, next slide. Now, this slide here is already something I've mentioned. I told you that we only have 20 amino acids. But then how can I make billions and billions of different kinds of polypeptides? It's the sequence that matters. And the sequence will then determine the overall shape, and the overall shape will determine the overall function. And that's what this line keeps stressing as well. A protein's three-dimension structure affects its specific function. Change the structure, change the function. And notice that's also a mantra that we had as one of our themes. So changing the structure can change the function. Okay, next slide. Here is a picture of an amino acid sequence. And then it's changed into some other version, and then finally some other version. So it actually folds and twists and changes shape. Now, one of the things you need to remember for this polypeptide sequence here, one end is always the amino group. And when I say amino, I'm talking about the NH2. The other end of this thread or string, the other end is the carboxyl group, which is the COOH. So if I put a lot of Lego blocks together, for example, the one end will always have the bumps, and the other end of this long chain of Legos, when you snap all the Legos together, will be the little grooves at the other end. So if you ever play with Legos, you know that the Lego blocks have a bump on one end and a groove on the other end. And if you put all the Legos together, snap them all, one end is always going to be the amino group, which is the NH2, and the other end will always be the carboxyl. Let me go back to a picture to show it to you. So one end will always be amino, the other end will always be carboxyl. And when you put them all together, notice again, when you make a long chain out of it, one end is always the amino group, the other end is always the carboxyl group. Okay. All right, next slide. All right, again, the author is now stressing the exact same example that I gave you before. It says, changing the structure of it will change its function. And that's exactly the same classic example that we always give about sickle cell anemia. Changing the structure of the red blood cell changes the function and the oxygen carrying ability. Okay, here's a picture of the same thing. Here is a normal hemoglobin, a normal polypeptide, gives you a normal donut red blood cell. Here is a one single chain. Now look at the difference here. There's only one letter difference, one amino acid difference. Nimish, man, is it that simple? Yes, you can screw up a structure of something by changing a single amino acid. So a single amino acid difference can make a change in the shape of the red blood cell. Our red blood cell and the chimpanzee's red blood cell is also that subtle. The one amino acid difference that makes a chimpanzee red blood cell versus a human red blood cell, it is that subtle of a difference. One amino acid difference. And notice it changes the shape of it. Okay, next slide. Okay, this is again stressing the same thing, so I'm not going to stress it over and over. But you do need to understand that there are some diseases out there that have to do with screwed up brain proteins. And I'm going to use this line right here to summarize this part. Prions. They're called prions. And so have you ever heard of mad cow disease? Well, mad cow disease is related to this. So normally we don't hear about proteins themselves being something that causes diseases. Usually it's a virus that causes a disease or a bacteria that causes a disease. Something that's typically a living thing that causes a disease, right? Usually that's what we think about, that either a bacteria or a virus, something living that can cause a disease. Well, in this case, it's something as a chemical like a prion, which is a kind of a protein. And it's a misfolded protein that forms in the brain because of mad cow disease, and then you get all kinds of problems. So I'm going to move on to the next slide for the picture. So here's what happens. Here's mad cow disease. Okay. And in humans, they have other names for that disease, but it can affect humans. So if you didn't know that, mad cow disease can affect humans. It causes trembling and shaking, and eventually you sleep and then don't wake up. So prions are very, very fatal. It actually causes brain damage. And that's why you get sleepy and sluggish and can't stand, can't walk, and have tremors because of prions causing mad cow disease. It was a big deal in McDonald's in England in the 80s. There was a outbreak of this bad disease in cows, and people that were eating at McDonald's in England, they came down with mad cow disease because it does spread from animal to animal by way of the meat. Okay, next slide. So here's a protein shape, very, very sensitive to the environment. And if you remember my two examples from the past, and one I repeated today even. So when you warm up an egg on a skillet, you're adding what? Temperature to it, right? You're frying it on a skillet. So but that means that temperature can affect the shape of a protein. The other example I gave in a previous video was enzymes in your mouth. The enzymes in your mouth work at pH 6. But once the enzymes are swallowed and the enzyme ends up in the stomach, where the pH of the stomach is pH 2, then the enzyme stops working because the protein is changed. So you can actually turn on and turn off a protein by changing the temperature or by changing the pH. And we call that a technical word that you're going to need to know, and that's called denaturation. So when you denature or denaturization, that's when you are changing the protein shape based on either temperature or pH. And you're actually unraveling, unfolding, and when you change the structure of that protein, you change its function. That's how you turn on or turn off an enzyme or turn off the function of a protein by either temperature or pH. And again, it is very sensitive to the environment. Okay, now our last section is dealing with nucleic acids, and that's your DNA, your RNA. So this slide right here, you should know the full form of DNA and RNA, and it's listed on this slide. So the full form of DNA and RNA. Now, DNA and RNA are made out of certain building blocks. Again, we're going to have our dehydration story for putting it together. So over here, we have our DNA. Now, when you put a lot of DNA pieces together, like the RNA or DNA pieces together, when we put the DNA pieces together, we form what's called a gene. Now, my next definition I'm going to give you is very important for now and for future chapters and exams. So here's my definition of a gene. A gene is a sequence of DNA that codes for a specific protein. I'll repeat that again. A gene is a sequence of DNA that codes for a specific protein. So when you have sickle cell anemia, for example, why do you not have a correct red blood cell made? Why are you making the sickle cell shape red blood cell? That's because the gene for that is messed up somewhere on that person's entire DNA strand. So gene is a sequence of DNA that codes for a specific protein. So you don't just come down with sickle cell anemia. Sickle cell anemia is a genetic disease. So somehow in that person, that gene is messed up And now it doesn't have the correct DNA sequence, therefore it makes a faulty protein, a faulty red blood cell. Okay, here's a slide you want to know for sure. This slide shows you a gene, which is a sequence of DNA that codes for a specific protein. The DNA has the information in this gene. So the gene is like the area that has the code for everything. And then the DNA gets converted to RNA, and then the RNA gets converted to a protein. We will see this picture again and again and again. So DNA is a sequence or rather, back up. A gene is a sequence of DNA that codes for a specific protein. That's where the code is, and then the DNA gets converted to RNA, and that gets converted to the protein that's made. So in sickle cell anemia, there must be something faulty right here in that DNA sequence, which gives you a faulty RNA, which gives you a faulty protein. Okay, next slide. Okay, now just like all the other nutrients, what do we call the building block for DNA? We call them nucleotides. So the basic building block of DNA is a nucleotide. Now instead of writing these three here, we're going to see them in a picture form on the next slide. So each nucleotide, which is the building block for DNA, has these three, and we're going to see them on the next slide. So here they are. We see that it has a sugar, and we call it a pentose sugar because it's a five-sided figure, like the Pentagon office, like the military has. So it's a five-sided pentagon kind of thing. And it's going to be a sugar, a pentose sugar. Then it has a phosphate group, and it has a nitrogenous base. Now the nitrogenous base can be A, G, C, and T. If you remember from a previous chapter, I believe it was the chapter 1 or it's chapter 1, I told you what pairs you have. You have A pairing with T and G pairing with C. So again, notice you're going to need to know that again. So A pairs with T, G pairs with C. And these are called nitrogenous bases. Okay. Next slide. Again, the author has given them to you as a list. So A is adenine, G is guanine, T is thymine, and C is cytosine. Make sure you know the names that go with the letters. So adenine with A, guanine with G, thymine with T, and cytosine with C. So you're going to need to know the names that go with the letters. Now, how do we put them together? That's the next slide. That's this. So we see that adenine has 2 rings, guanine has 2 rings, but thymine has 1, and cytosine has 1. Now remember how I said that there's a certain pairing that A pairs with T? So you can see here that a 2-ring structure, A, pairs with a 1-ring structure, T. That's why A pairs with T. So something that has 2 rings will pair with something that has 1 ring, A and T. Same is true for G and C. G, or guanine, has 2 rings, and cytosine, C, has only 1 ring. So only a 2-ring structure pairs with a 1 ring. You cannot have 2-ring pair with 2 rings or 1 ring pair with 1 ring. It wouldn't work. And we'll see that as to why in another chapter. But this DNA model here, this DNA model, is because A, which has 2 rings, pairs with T that has 1 ring, and guanine that has 2 rings pairs with C, which has cytosine with 1 ring. So 2 rings pairs with 1 ring. All right, so be aware of that. Next slide. Okay, again, you're not writing this part. I already gave it to you. We are doing dehydration to put things together. Now let me go back for a slide. When I join A and T together, it's going to be done with 1 single hydrogen bond. So one single hydrogen bond. Whereas guanine and cytosine, we'll see later, has 3 different bonds. And I'll show you a picture of that later. Okay, next slide. Okay, this slide says something I've already shared with you a couple times. A only pairs with T, G only pairs with C. So we've already said that, simply a slide showing you the same. Here is the same picture again. We see G pairing with C, A pairing with T. And notice it's done with hydrogen bonding, and that's what I want you to know now. Now later on, we'll see a more clarified picture talking about the single and the triple bonding, but for right now, stick with this slide where it says it's a hydrogen bonding that pairs A with T and G with C. And we'll see later how many of those hydrogen bonds apply in a better picture. Next slide. Okay. Now we have something called RNA. Now RNA has 3 differences. You will easily be asked about these 3 differences when it comes to either my lecture exam or the department exam. So here's how it works. One difference is the sugar. It doesn't have deoxyribose. Let me go back to the picture. See how this one says deoxyribose? That's for DNA. See how DNA says deoxyribose? Well, that's not true for RNA. RNA, there it is, RNA uses something called ribose sugar. So that's your first difference shown here. So RNA uses a ribose sugar, not deoxyribose. The other difference is instead of thymine, RNA will use uracil. So RNA will use uracil, U. So for RNA, it won't be A and T. It's going to be A and U. Okay, let me say that again. So DNA had A pairing with T. But since RNA does not have thymine, instead it has uracil, it's going to be A pairing with T when it comes to RNA. Then the last difference is RNA is single-stranded. So if you look in the picture here, we see that it's double-stranded. Okay, there's 2 strands. Here's another picture showing you the same thing. The DNA is double-stranded. Well, that's not true for RNA. RNA is single-stranded. Okay, next slide. This shows you the differences. So again, for RNA, it's going to be a ribose sugar, and instead of T, it's going to be U for uracil. So the author does have pictures for you. Next slide. Again, that's a repeat. Okay, don't care. Okay, don't care. Went too far, so let me backtrack. Okay, don't care, don't care, don't care. Okay, and this one I do care. Now remember I told you that some people don't have the enzyme lactase, and I mentioned Asians typically have less of it. Well, if you have a DNA somewhere in your body, and here is a gene here, it's how it says lactase gene. If that is missing or that is not working, then you are not going to make the enzyme called lactase that's shown at the top. Or it may be a faulty one, right? So you're either not going to make it or you're going to make a faulty one, and therefore you're going to have what's called lactose intolerance because you don't have an enzyme to break down the milk sugar. So when you take dairy foods and lactose is in them and you're not breaking it down, it becomes food for bacteria in your gut, causes gas, bloating, diarrhea, and all of that. So lactase is the enzyme that breaks down lactose, and in some people with a genetic problem where they don't make enough or they make a faulty enzyme, then that's a problem. Okay, next slide. And I wasn't kidding when I said Asians. You can see here 90% of Asians lack this ability to make the proper lactase enzyme, and that's why I gave it that to you. Don't care. Don't care. And that is the end of the chapter. So now you're ready to move to chapter 4.